Hello everyone, my name is Aya Trebec and today I will guide you through an internship project I did back in 2017 at the Marine Biology Station of Piran with the National Institute of Biology in Slovenia. The project lasted 1 month, through which I focused on the cytological and stress effects on Mediterranean mussels. The warming and cooling of the Earth have been occurring with oscillating frequency through eras. Although global warming is a natural process to some extent, the most concerning thing is that with anthropogenic impacts, it is occurring with a much faster rate than naturally. In fact, it is possible to observe from this graph that the sea surface temperature in the Mediterranean has increased for almost 1 degree Celsius from 1990 to 2015, and it is predicted that the temperature will rise for 0.4 degrees Celsius every decade, with a 1.2 degrees Celsius rise by 2050, which could imply potential negative effects on the marine organisms from the cellular to the ecosystem functioning. An important bioindicator species used in ecotoxicological studies is the Mediterranean mussel, also known by the Latin name Mytilus galoprovincialis, and so you can see it here on this picture, which is a tolerant bivalve species that lives in the intertidal rocky areas, as well as attached to buoys and boats. I decided to use Mediterranean mussels first because they are easy to collect and monitor, and second to examine how these organisms respond to different seawater temperatures. Throughout the project, I used 3 biomarkers, but for today, I will focus on just one, which is the neutral red. It was used to find out if it could be the indicator for metabolic changes, which occur when mussels are exposed to stressful factors, in this case, elevated or low seawater temperatures. But before moving on, I would like to explain the bottom right photo, as well as what is the neutral red retention assay. Lysosomes are organelles that are dealing with nutrition, tissue repair, and cellular components turnover. They can be found in the granular hemocytes and are important for destroying pathogens and exhibit immune response by releasing lytic enzymes into the cytosol. As we can see from this photo over here, this is the hemocyte, which is a cell with a nucleus that is not colored, and these orange and red dots are separate lysosomes colored by neutral red dye. So neutral red retention assay is a broad spectrum biomarker. Through this ecotoxicological method, it is possible to measure the membrane structural integrity of lysosomes, where a long exposure time reveals that the lysosomes have a structural healthy membrane, whereas a short exposure time means damaged membranes because of the presence of stress factors in the environment. Therefore, the destabilization of lysosomes occurs due to environmental or chemical stress and is determined by the secretion of the neutral red into the cytosol. The lysosomes can as well get bigger and fuse together. But let's move to the methods. Mediterranean mussels were collected in August of 2017 from a buoy 2 meters below the sea surface in front of the institute. They were placed in a tank with an inlet and a drainage pipe that allowed the collected mussels to be constantly supplied with fresh seawater. Mussels were let acclimated for 2 days, and after that 2 plastic buckets were filled with seawater and 5 mussels were placed in each one. Specimens of each bucket were exposed to different temperatures. One bucket was with cold water at 18 degrees, and the other one was with hot water at 29 degrees Celsius. A water pump was put into each bucket so that the water temperature was evenly distributed through the whole water volume, and seawater was changed every 2 days just to keep the culture fresh. What I needed for the neutral red retention assay was the hemolymph of the mussels, which contains hemocytes. As a control, I extracted the hemolymph from muscles that were not exposed to any temperature differences. So, for extraction of the hemocytes, muscle valves were opened with scissors and kept opened with a pipette plastic needle. Seawater was let out. So it looked something like this. And this white part that you can see is the posterior adductor muscle, which looks like this when it is cut with scissors. And hemolymph was taken from the posterior adductor muscle, as I said, with a 1 milliliter syringe and 21 G needle. 0.1 milliliters of hemolymph were extracted along with the same quantity of physiological saline solution, adjusted with the adjusted pH to 7.4, which is the pH of the seawater found in front of the institute. The needle was then taken off the syringe to prevent hemocytes from damage, and the syringe content was put into an Eppendorf tube, marked and put into ice. Afterwards, slides covered with poly-L lysine solution were used for better adhesion of cells. 50 microliters of muscle hemolymph were applied to the slide, which was placed into humidity chamber for 15 minutes. And right before the time ran out, I prepared the neutral red working solution and added 40 microliters to the place where cells adhered. Cells were placed into light-proof humidity chamber for another 10 minutes. Then they were taken out of the chamber and I covered them with a glass cover slip and wiped the excess liquid off the edges with a cotton paper. The slides were examined every 15 minutes for the first hour and then every 30 minutes for another hour and a half under the microscope. Here we have hemocytes from different conditions. So the little orange and red colored circles are the lysosomes that contain the neutral red biomarker, but we saw them previously in the introduction slide. As you can see, at 90 minutes, more hemocytes that did not release dye into the cytosol were observed in muscle samples of the cold seawater bath, whereas those exposed to hot seawater bath had percentually decreased. And this is an indicator of more stressful conditions. Since lysosomes were stressed by the temperature, the membrane integrity was destabilized faster. In addition, I noticed that the release of neutral red dye and membrane fusion occurred in a shorter exposure time in muscle samples taken from the hot seawater bath, because already at 45 minutes, the lysosomes appeared damaged or bigger, whereas for the muscles exposed to cold seawater, slight changes in the lysosomal structure were observed after 90 minutes. And this in particular can be observed here from this cell, where the lysosomes are not so evident, and this indicates that the neutral red was released into the cytosol. For this particular observation, I have hypothesized that the microscope light passing through the slide might have warmed the samples, as well as the room temperature was not adjusted to 18 degrees Celsius. One very important thing that you can observe here is the similarity between hemocytes of the control, this one, and the hot seawater bath after 90 minutes exposure. But why? Because muscles were collected in August with the particularity that the seawater in August was 27 degrees Celsius. Sorry, the temperature of the seawater was 27 degrees Celsius. And in both cases, indeed, it is possible to see bigger lysosomes and in some cases even lysosomes fused together. So an example of lysosomes fused together is this one as well as this one, and bigger lysosomes are these that are still a bit smaller than the fused lysosome. Lastly, for this project, I learned that global warming is inevitable and unfortunately it has negative effects even on the more tolerant marine species. However, I do believe that we humans have the potential and intelligence to avoid what is so apocalyptically presented on this photo here. We have the ability to change our daily habits and reduce our footprint in order to move towards a better future and allow future generations to thrive. One of the possible solutions that could potentially help maintain the fisheries economy could be the establishment of submerged cages in the open sea. These cages are used for aquaculture purposes and designed especially for rough seas. The open sea environment is generally characterized by colder water masses in comparison to the coastal marine ecosystems. So, just to have a clearer idea, mussels could be reared in colder waters with relatively strong hydrodynamic characteristics and a constant current flow, which would supply the bivalves with food. And with this, I would like to thank you for the attention. I would also like to thank my mentor, Andrea Ramšak, at the National Institute of Biology in Slovenia and the Polytechnic University of Marke, who allowed me to do the internship abroad. A special thanks goes to the organizer of the National Marine Science Students Conference. If you have any questions at all, go ahead. I will be more than happy to answer. And you can also contact me via email that is right here at the bottom. Here are some references if you're interested. I use this for the presentation. And yes, thank you very much.